Welcome to a tour of auxin metabolism, where the plant hormone indole-3 acetic acid, or IAA, shapes growth and form. This presentation traces how IAA is made, moved, stored, and broken down to create precise concentration gradients in tissues, and why that spatial control is so crucial for development from roots to flowers. Let's explore the metabolic network that underlies those gradients. Plants must precisely control auxin levels because development depends on sharp chemical gradients, not random hormone distribution. Too much or too little IAA in the wrong place causes big problems, so plants combine directional transport with local metabolism to place auxin where it's needed and remove it where it's not, tuning growth in response to internal and external cues. The auxin story starts with the aromatic amino acid L-tryptophan, produced via the chloroplast shikimate pathway and acting as the central precursor for IAA biosynthesis. From tryptophan, multiple parallel routes branch out and converge, with key intermediates like IPYA, IAOX, and IM feeding into IAA production, highlighting that local, organ-specific synthesis is a major feature of auxin control. The dominant IAA biosynthetic route is the IPYA pathway, conserved across land plants. First, tryptophan is deaminated to indole-3 pyruvic acid by TAA/TAR enzymes. Then IPYA is irreversibly decarboxylated by yucca family monooxygenases to produce IAA. This two-step route includes a committed, rate-limiting step that defines the main production line for active auxin. When the main IPYA production line is disrupted, plants reveal how essential it is. Single mutants are often mild due to redundancy, but removing multiple TAA/TAR or YUC genes causes severe developmental defects. Mutant plants show altered leaf and flower patterns, aberrant vasculature, reduced stature, and often lack a primary root, demonstrating that this pathway is critical for normal growth. Plants retain backup options. Several alternative routes contribute to the auxin pool even if they play smaller or conditional roles. These include the IAOX pathway active in some species, pathways using IM intermediates, and routes converting indole-3 butyric acid into IAA, each adding redundancy that can become important during stress or in particular tissues. To maintain precise auxin gradients, biosynthesis must be balanced by active inactivation, creating a dual system of reversible storage and irreversible catabolism. Reversible conjugation provides a ready pool of inactive auxin that can be liberated quickly, while irreversible pathways permanently remove excess auxin to create minima required for specific developmental events. Plants store IAA by conjugating it to sugars, amino acids, or methyl groups, temporarily inactivating the hormone for later reuse. The most common storage form is IAA glucose made by UDP glycosyl transferases. Amino acid conjugates formed by GH3-type enzymes can be hydrolyzed back to IAA, and methylation creates nonpolar forms that affect transport and local gradients when converted back. Excess auxin is permanently disposed of through irreversible catabolic routes. The major pathway oxidizes IAA to OX-IAA via DAO-type dioxygenases, often followed by glycosylation for final disposal, while specific conjugation reactions produce catabolites like IAASP and IAA glue that are not hydrolyzed back, ensuring robust removal of surplus hormone when needed. Where metabolism happens is as important as how much is made. Spatiotemporal expression of biosynthetic and inactivation genes sculpts local auxin maxima and minima. YUC genes create local peaks driving organ formation, TAA family members set up root gradients for stem cell niches, and GH3 enzymes define minima in specific domains, so location and timing control developmental outcomes. A focused case study shows that proper root development depends on auxin produced within the root itself. Coordinated TAA and YUC expression patterns in embryos set the apical basal axis. Local biosynthesis in the meristem maintains stem cells even without long-distance transport, and regeneration after wounding relies on local IAA production near the cut site. Auxin homeostasis is orchestrated by many regulatory layers working together. Metabolic feedback loops adjust synthesis and degradation, hormonal crosstalk modulates inputs, transcriptional networks switch metabolic genes on and off, epigenetic marks set long-term potentials, and post-translational or subcellular controls fine-tune enzyme activity and localization for integrated responses. Transcription factors and epigenetic mechanisms act as a command center, directly tuning auxin metabolic genes in response to development and environment. For example, heat-activated factors can upregulate YUC and TAA genes, repressor complexes recruit histone marks to silence YUCs during floral patterning, and DNA methylation pathways can stably silence specific biosynthetic loci, shaping expression patterns over time. The final layer of regulation comes after transcription. Enzymes are switched on or off by phosphorylation and other modifications, degradation machinery can be activated by substrate presence, and subcellular partitioning separates production, storage, and signaling. Transporters in the ER and plasma membrane move auxin between compartments, creating local concentrations that determine signaling output.